Nucleic acids are essential molecules found in the nucleus of a cell, and they come in two types: DNA and RNA. They are crucial for making RNA and proteins, which are vital for the functioning of living things. The main difference between prokaryotic and eukaryotic DNA is that prokaryotic DNA is double-stranded and circular, while eukaryotic DNA is double-stranded and linear. The modern understanding of DNA has evolved from the discovery of nucleic acid to the development of the double helix model. In 1869, Friedrich Miescher began working with white blood cells and discovered a unique chemical substance called nuclein. Nuclein, which has high phosphorus content and acidic properties, was later named nucleic acid. By the early 1900s, it was known that nuclein was a mix of proteins and nucleic acids, leading to the identification of DNA and RNA as the two kinds of nucleic acids. The length of DNA in a typical mammalian cell is approximately 2.2 meters, which is condensed and coiled to fit into a small nucleus. This packaging is essential for DNA to be accommodated in such a small space. DNA is structured in a double helix with major and minor grooves. It is further organized into nucleosomes, chromatin fibers, and eventually into chromosomes during cell division. In prokaryotes, DNA is packaged within a nucleoid, which is not enclosed by a nuclear membrane. The DNA is circular and interacts with various proteins to form a compact structure. Prokaryotic cells, like E. coli, have a nucleoid that is highly folded and contains a naked ring of DNA. This DNA is approximately 1,100 micrometers long when stretched out, but it is compacted to fit within the small cell. The compacting of the 1,100 micrometers long nucleoid involves coiling and supercoiling, assisted by RNA connectors and proteins like Hub proteins, DNA gyrase, and DNA topoisomerase, maintaining the supercoiled state. DNA packaging in prokaryotes involves multiple stages, including the formation of loops and supercoiled structures, which are further compacted to fit within the cell. Eukaryotic cells have well-organized nuclei with DNA associated with histone and non-histone proteins. This organization is more complex compared to prokaryotes. Histones are proteins rich in lysine and arginine residues, which are positively charged and help in organizing DNA into nucleosomes. These histones form an octamer around which DNA is wrapped. Negatively charged DNA wraps around positively charged histone octamers to form nucleosomes. Each nucleosome consists of histone proteins H2A, H2B, H3, and H4, with H1 binding the DNA thread. Each nucleosome contains about 200 base pairs of DNA, with 146 base pairs wrapped around the histone core. Nucleosomes are the repeating units of chromatin, which appear as beads on string under an electron microscope. Nucleosomes coil to form a solenoid structure, which further supercoils to form chromatin fibers. These fibers condense at metaphase to form chromosomes. The packaging of chromatin at higher levels requires additional proteins called non-histone chromosomal proteins, NHC, which contribute to the organization of chromatin. DNA packaging progresses from naked DNA to nucleosomes, chromatosomes, solenoids, 30 nanometers fibers, and finally to chromosomes during cell division. Heterochromatin is more condensed, gene-poor, and stains darker, while euchromatin is less condensed, gene-rich, and stains lighter. These regions have different roles in gene expression. Chromosomes have distinct parts, including telomeres, heterochromatin, centromeres, and euchromatin, each with specific functions in maintaining chromosome integrity and gene expression. By the early 1900s, geneticists knew that genes control inheritance and are located on chromosomes, which are composed of DNA and proteins. Initially, proteins were thought to be the genetic material due to their complexity. Over time, it was discovered that DNA, not protein, is the genetic material. DNA molecules are large and vary significantly, leading to the understanding that DNA is responsible for genetic variation and inheritance. In 1928, British medical officer Frederick Griffith conducted an experiment on the bacterium Streptococcus pneumoniae, which causes pneumonia in humans and other mammals. Griffith used 2 strains of Streptococcus to find a cure for pneumonia: a virulent, smooth, and encapsulated S-type, and a non-virulent, rough, non-encapsulated R-type. Griffith conducted 4 experiments on these bacteria. First, he injected R-strain bacteria into mice, which survived because it did not cause pneumonia. Second, he injected S-strain bacteria, which caused pneumonia and killed the mice. In the third experiment, he injected heat-killed S-strain bacteria into mice, which survived. Finally, he mixed heat-killed S-strain with live R-strain bacteria and injected it into mice, which died. Griffith recovered live S-strain bacteria from the dead mice. Griffith's experiments showed that harmless R-strain bacteria could be transformed into deadly S-strain bacteria. He suggested that genetic material from the heat-killed S-strain had changed the living R-strain into S-strain. This led to the conclusion that a transforming principle from the S-strain was responsible for this change. In 1944, Oswald T. Avery, Colin M. MacLeod, and Maclyn McCarty conducted experiments to prove that DNA is the genetic material. They purified DNA, RNA, proteins, and other materials from S cells and mixed them with R cells. Only DNA was able to transform harmless R strain into virulent S strain. They discovered that protein digesting enzymes and RNA digesting enzymes did not affect transformation, but DNA digesting enzymes did. This proved that DNA is the transforming principle. Further experiments by Alfred Hershey and Martha Chase in 1952 confirmed that DNA is the genetic material, not proteins. This diagram illustrates how DNA transforms bacteria. Rough non-virulent strain R, when combined with protease, does not transform and the mouse dies. However, when combined with DNase, the rough strain transforms into a smooth virulent strain, and the mouse survives. Hershey and Chase worked with viruses that infect bacteria, known as bacteriophages, which are composed of DNA and protein. They used radioactive phosphorus for DNA and radioactive sulfur for protein. They grew these viruses on a medium containing the radioactive elements. In the first experiment, they tested proteins and found that bacteria infected with protein-labeled viruses did not show radioactivity inside the cells. In the second experiment, they tested DNA and found that bacteria infected with DNA-labeled viruses showed radioactivity inside the cells. This concluded that DNA is the genetic material. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA, but not radioactive proteins. Similarly, viruses grown with radioactive sulfur contained radioactive proteins, but not radioactive DNA. Radioactive phages were allowed to infect E. coli, and the viral coats were removed using a centrifuge. Bacteria infected by viruses with radioactive DNA were radioactive, indicating that DNA was transferred from the viruses to the bacteria. Bacteria infected by viruses with radioactive protein were not radioactive, indicating that proteins did not enter the bacteria. This confirmed that DNA is the genetic material passed from viruses to bacteria.